Hello and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Professor Kim Wolff. Listeners, Kim is Director of King's Forensics and Head of the Drug Control Centre, which is the only WADA-Accredited laboratory for sports testing in the United Kingdom. She is also Director of the London Athlete Passport Management Unit, or APMU, and she was named a member of the Most Excellent Order of the British Empire, or MBE, for her services to road safety. And today, listeners, we are going to talk more about Kim's research as well as her roles leading a WADA-accredited lab in APMU and more. So, Kim, welcome to our show today. How are you?
I'm very well, and thank you so much for inviting me to talk to everybody today.
Well, we are thrilled to have you with us. And for listeners who don't know you yet, Kim, can you start by telling us more about yourself and perhaps how you originally got interested in this area of forensics and anti-doping science?
Okay, well, thank you. Yes, I originally started out studying sports science, actually, and was even a doping control officer at one point in my career. But I really began to develop an interest in drug testing and analysis as part of my PhD. This was funded by the Medical Research Council and was concerned with drugs of abuse and studying the pharmacokinetics of methadone in drug addicts. This took me into the laboratory and the use of HPLC and gas chromatography. Involvement with forensic science came next, and as a result of my being invited to sit on the Secretary for State for Transport's expert panel on drink and drug driving, and also some research I was doing at the time. I was asked to chair the expert panel on drug driving and was integrally involved in bringing new legislation for drug driving to England, Wales and Northern Ireland. When the role of Director of King's Forensics came up, it seemed an obvious fit for me, linking analytical science and forensic science. But I think anti-doping is a brilliant field to be in, never a dull moment, and of course being front and centre of the efforts in the UK to maintain the integrity of elite sport.
So I know, Kim, we mentioned a couple of different titles in our introduction there. It's wonderful to hear about how you got to these roles, but perhaps could you share next what your responsibilities are in each of these different positions?
Okay, well, I'm Director of King's Forensics at King's College London, which is a small academic unit that encompasses ISO-accredited laboratories, the Forensic Genetics Laboratory, and an ISO-accredited City of London Police finger mark laboratory, as well as the Drug Control Centre. We have research ongoing in trace analysis related to explosives and scenes of crime work. Another avenue of research is wildlife forensics, looking to improve crime detection there, as well as bioanalysis for drugs of abuse. We also carry out research in wastewater analysis, and I oversee a mass spectrometry facility for clinical trials work. We also do education, as you might expect. Then we run 2 master's degrees and teach some undergraduate modules in forensic science. So lots going on. The DCC, of course, sits within King's Forensics, and I'm the laboratory director of this anti-doping laboratory with overall responsibility for ensuring the team at the Drug Control Centre, there are about 30 of us, maintain the quality standards embedded in the international standard for laboratories, and of course strive to deliver testing results within the WADA-framed timeframe. We also look to innovate and keep abreast of developments in the field.
And I know we talked about the Drug Control Centre, and this is a really cool type of position to work in these WADA-accredited laboratories. So can you tell us a little bit more, Kim, about the history of the Drug Control Centre there in the UK and maybe share some of the things that it does particularly well?
Well, the Drug Control Centre is a very well-established anti-doping laboratory, and I inherited a lab that has been around since 1984, and it was one of the first laboratories to be accredited by the then International Olympic Committee. The Drug Control Centre has received continuous accreditation since then, which is a very proud heritage. Our work in the analysis of peptide hormones is well known, and we are one of only a handful of labs to include the Endocrine Module as part of our APMU. We do growth hormone bioanalysis well, and the analysis of anabolic steroids. I would say that we do everything well because that's a requirement of being an anti-doping laboratory. We have a good research ethics, I think, being based in the university, and like to keep abreast of everything that's ongoing in anti-doping science as well.
Definitely. And I think that last point that you made is huge. Anti-doping is this dynamic field where there's constantly new research coming out that needs to be integrated into the laboratory. So can you talk about what are maybe some of the biggest challenges of running a WADA-accredited laboratory in this dynamic anti-doping environment?
I think there are many challenges because ultimately the work we do has the potential to impact on the reputation and livelihood of an elite athlete. Maintaining quality standards in a busy laboratory with a throughput of about 12,000 samples a year is a given, but it is a challenge. Staying ahead of those that cheat is our biggest challenge. And we must not forget that it's rarely an individual athlete who is at fault on his or her own, but a team who conspire to enable the athlete to gain an unfair advantage over other athletes. Doping has become sophisticated, and keeping ahead of the curve in that regard is everyone's challenge, I believe.
Definitely. And I'm really glad you brought ahead this point of staying ahead of the cheaters. This is something I hear people talk about a lot, but I think it's a difficult problem to solve. So perhaps a follow-up question, Kim, if I can. How does the lab stay ahead of the cheats? And how do you kind of take these steps to make sure you're on that cutting edge?
Well, I think there's an awful lot of collaboration. We talk to other anti-doping laboratories. We are well informed by WADA and anti-doping organizations such as UCAD based here in the UK. I think there's a lot of communication, a lot of sharing of intelligence, and a lot of working together to try and address this challenge.
Absolutely. And a lot of working in general, I think. Now, we also mentioned that you lead the London APMU, that Athlete Passport Management Unit. And this might be a term that some listeners have heard and perhaps some listeners have not. So can you maybe first just define what this is and then tell us more about your APMU and how it's supporting clean sport?
Well, the Athlete Passport Management Unit here in the UK is one of only 16 across the anti-doping world, and it monitors individual athletes on a longitudinal basis. So it supports clean sports by monitoring steroidal, hematological, and lastly, endocrinological biomarkers of an individual over time. This means that we measure endogenous steroid hormones, biomarkers of growth hormone, and blood parameters according to a norm that's established for the individual athlete. It's therefore possible to monitor the individual for the duration of his or her career as an elite athlete, which is more meaningful perhaps than randomly collected urine samples.
Definitely. And Kim, I think you mentioned earlier that you're one of the few units that are able to monitor the endocrine module. Can you comment on what this has been like?
Well, it really has just begun. We, along with other laboratories, have introduced this testing, again, to augment and supplement other longitudinal testing programs. And it's really geared at the identification of abuse of growth hormone and its analogs. So that's added to our repertoire of tests that we look at over time with individual athletes.
That makes sense. And how does that work in terms of adding one of these additional modules to the lab? Did you have to kind of restructure things or grow the staff, or did it fit pretty well within the infrastructure that you had already?
Well, it fitted pretty well, I guess, with what we were doing already, because we're already testing for biomarkers. So it was a matter of developing technology and methodology to support the new tests. And also WADA's ADAMS monitoring or platform for reporting results has also been developed to enable us to report endocrine test results in the same way that we would do for steroid and hematological. So I think the system is, although in its infancy, it is growing very nicely.
Definitely. And Kim, I know the work that you do spans a variety of different areas in forensic toxicology as well as in anti-doping science. So can you talk about perhaps how your background in this area of forensics, or perhaps even going back further in sports science, helps you in the work that you're doing today in anti-doping?
Well, my background has really been embedded in analytical science, measuring biomarkers of illicit substances. And my early research was concentrated on the analysis of methadone in biological fluids using chromatographic techniques. I became involved in work for the government on drug driving, as mentioned previously, and that involves setting thresholds for illicit and prescribed psychoactive drugs. And I think this work has really helped my transition into the world of anti-doping science, since there are, as we know, threshold substances within the WADA prohibited list, although I have to say very many more compounds than in the UK drug driving legislation. I think the need to ensure integrity of the analytical process through UCAS accreditation in forensic work is very similar to that required for anti-doping. So that was a good foundation, if you like. Anti-doping and forensic toxicology have very many similarities in that regard.
Absolutely. I appreciate you talking about some of these similarities. Maybe I'll flip the question a little bit and ask, what have been some of the differences maybe that are unique to anti-doping compared to forensic toxicology and some of the other work that you'd done previously?
I think what is unique to anti-doping is the wide breadth of compounds which are present on the WADA prohibited list. This is a big undertaking for laboratories to screen for all of those substances and also to be able to confirm them when a suspect is identified. I think this is probably quite different to forensic toxicology, which people may disagree, has a smaller repertoire of compounds to look at over time. I think we also have tight reporting deadlines, which is different from forensic toxicology. And it isn't criminal work in most cases. So we're working with anti-doping organizations, WADA, when we identify a suspect. So the mechanism for dealing with positive test results is a little bit different as well.
Well, Kim, sounds like you are doing some really exciting work. And I like how you highlighted, some of the overlaps and some of the differences between anti-doping and some of the other research that you're doing. But for listeners out there who aren't familiar with just the breadth of the work that you do, can you give us an overview of maybe the range of different kinds of research questions that you've worked on and some of your big contributions to the field?
So my early research was concerned with whether or not we could carry out therapeutic drug monitoring in patients prescribed methadone. My pharmacokinetic studies with addicts prescribed the drug were the first to be carried out in the UK. I introduced the concept of auto-induction of methadone kinetics and showed the risk of overdose if the onset of daily dosing with methadone happened too quickly. I also carried out some seminal work with pregnant drug addicts and showed that the clearance of methadone increased during pregnancy, requiring dosing support in the third trimester. Later on, I was funded by the Department for Transport to establish a biomarker suitable for identifying continuous drinking in drivers applying for a driving license having been disqualified. My research compared liver function tests and gamma-GT with carbohydrate deficient transferrin, and I was able to show that percent CDT was a more accurate biomarker of continuous drinking than other tests. My results were taken up by the Department for Transport, and CDT was used as a sole marker to assess drinking in drivers as a consequence of my research.
I think that's wonderful. And I think being able to have these impacts, both kind of on the clinical side, in the sort of policy side for this drunk driving aspect, and then also the anti-doping, obviously those immediate impacts in sport. What has it been like for you to be able to sort of see the fruits of your labor having an impact, Kim?
It's been rewarding to be able to carry out research that can then be applied in a practical sense. I think it's helpful when research has that kind of application, and I think it enables policymakers and organizations to see the value of undertaking scientific research. So it's been a good experience for me, and I think it's been valuable in the communities where my research has had an impact.
Definitely. And I'd love to dig into some detail, if we can, on your work on biological markers of drug misuse. You've been working in this area, Kim, for more than 20 years. So can you tell us a little bit more about what this field looked like 20 years ago, and then perhaps how the field has changed or evolved over the course of your career?
Yes, I think it has changed quite significantly. I guess when I started out, there was very much drug users and alcohol users, and possibly a fairly small field of illicit drugs, which we know very well today, like your heroines and cocaines and amphetamines. I think the biggest single change today has been the introduction onto the illicit market of synthetic drugs that mimic the effects of our naturally occurring or endogenous compounds. We now have third-generation synthetic cannabinoids, synthetic opioids, not only the fentanyls, but also the nitrazines, synthetic benzodiazepines, and hallucinogens. So the picture is very much more complex than it was 20 years ago. I think this has been a challenge going forward for all laboratories involved in drug testing. Although many of the synthetic drugs are not predominantly used across all communities, and I would say not commonplace in sports. I also think the chromatographic instrumentation has also changed significantly from when I started out there. There's been significant improvement in the sensitivity of the instruments and the ability to monitor or detect a much wider range of substances. Keeping abreast, however, of this widening repertoire of drugs and understanding trends so that the laboratory effort is efficient and fit for purpose, I think is a constant battle and an important area that we need to keep abreast of.
Certainly. And have you seen growth in terms of the amount of research being conducted in this area?
I think there's certainly a good amount of interest in the detection of new psychoactive substances, as they're called, even though laboratories still, I think, struggle to keep up with the enormous number of drugs that have come onto the market in the last 5 or 6 or 10 years regarding new psychoactive substances and where to put their resources, which class of drugs should we concentrate on, which communities are we specifically interested in or able to provide a testing service for. So, as I say, I think the arena has become very much more complicated than it was before, and we have polysubstance users who use alcohol and illicit drugs and prescribed medications. So identifying where the different subpopulations of users fit in our communities, I think, is a challenge.
That makes sense. And when you look at the landscape of this biological markers of drug misuse research area, Kim, what do you see as perhaps the biggest areas of opportunity or perhaps the most pressing unanswered questions going forward?
Well, I think the biggest opportunity, certainly for the Drug Control Centre in terms of research, is the fact that as part of King's College London, we have access to what is known as King's Health Partners, which are all the teaching hospitals linked to King's College. And for collaborative purposes and joint research endeavours, this is a big opportunity. I think having access to clinical populations and clinical colleagues brings the potential of investigating some of the prescribed medications that are also abused by elite athletes. I think there is a lack of scientific information on the elimination or urinary excretion of drugs included in the WADA-Prohibited list. For example, the glucocorticoids such as prednisolone and the beta-2 agonists, salbutamol, or even the stimulants like methylphenidate. And this information, if it's there, may be old and has been undertaken without the benefits of the sophisticated instruments that we have at our disposal today. So I think I would like to see some consolidated work in this area and the development of those collaborative relationships.
Definitely. And I think another big area of research or important area of research, particularly for anti-doping science, is trying to better detect some of these substances like anabolic steroids that are produced naturally in our bodies, but athletes may also administer exogenously from other sources to try to improve their performance. So I know this is an area that your lab is working on, Kim. Can you give us a little bit more detail about your lab's ongoing work in this area?
Yes, of course. The detection of exogenous steroids is a concern for all anti-doping laboratories, and we, like others, are keen to develop methodology that helps differentiate between exogenous and endogenous steroids. We are building a research team here, as you said, and we have Dr. Aguilera as an expert in steroid analysis, who sits on the WADA expert group for gas chromatography combustion, IRMS, and he is researching at the moment the steroids mass balance and contributions to the steroid adaptive model, which is really looking at the metabolites of exogenous steroids and how they can be interpreted to understand drug use behaviors in our elite athletes. We also have a newly appointed expert, Fabio de Olivier, from the Brazilian lab, who's been tasked with developing methods to investigate long-term metabolites of anabolic steroids, using, again, the IRMS technique, and also some work to explore 19-NA metabolism. We also have links with Imperial University to look at steroid use in gym attendees. So watch this space. There's a lot that we're planning to do and going on in this research area.
Definitely. And I think in highlighting some of the active areas of research, you brought up a good point. And this is, I want to perhaps particularly dig into this idea of long-term metabolites. I think a lot of people may struggle to understand sort of why we still don't have a solution to this problem of detecting exogenous anabolic steroid administration. But it's because in these cases, even if you know what the metabolites are and identify some metabolites that are unique to these exogenous anabolic steroids, they may not stay around for a long time. So what is the timescale that you're looking at with some of these longer term metabolites?
Well, some of the metabolites last for a reasonably long time. So we're working actually with Imperial University to look at gym users because they take exogenous steroids willingly and then become chronic users and then struggle to stop using them. So we're looking at the cessation of steroid use in this particular group as a means to understand exactly how long the long-term metabolites are around in the system and can be found in urine. So I think because there are many different anabolic steroids which are produced exogenously, this is complicated work. And I don't think there is a standard in terms of the metabolites that we could address. So we're trying to look at a group of gym users and multiple substances to try and shed some light on long-term metabolites, how long they last in the system, actually, as you said, and which metabolites can be expected to be seen by particular exogenous steroids.
I think this is a really interesting area of research. And I think, Kim, when you're talking about it, you can feel the excitement. But is there another research project that you and the team are involved in right now that you think is particularly promising or exciting?
There are a few. I have a PhD student who has developed an assay to detect ethyl glucuronide, a biomarker of alcohol use in latent fingerprints, which could be used to detect binge drinking, so drinking excessively over a short period of time. We're also looking at the cannabinoids, so analytically both synthetic compounds, natural cannabinoids such as CBD and THC, as well as the endocannabinoids. So there's a lot going on in the cannabis space as well. And more closer to home in anti-doping, we're developing a portfolio of work using dry blood spots, concentrating initially on common steroids, but also looking at steroid esters and working to include IGF-1, which is a biomarker of growth hormone use as well. So that's an exciting area of work that's specifically related to anti-doping.
Wonderful. It sounds like you have a lot of really cool projects going on. And I'd like to perhaps touch just a little bit on this latent fingerprints idea. I think this is something that you use a lot in forensics. Listeners might not be familiar with it. So latent fingerprints are just fingerprints that are left on different things. And this is something that hasn't really been used in anti-doping at all. Is this something that you see will ever have potential, you think, in having any sort of anti-doping applications, or is it primarily just on the forensic side?
I think if there are sports where, in this application, where the sports governing body or the event organizers need to know whether a person has been using alcohol in competition, then latent fingerprints would be your go-to matrix because it can be done very simply, very quickly. And with the immunoassay tests that we've developed, the results are visible within 10 to 15 minutes. So it really is a sort of here and now test, if you like, to identify whether alcohol has been used or not recently. So if there was a need for that type of test in sports testing, then I think latent fingerprints would be quite helpful in that regard.
Certainly. And in terms of analyzing these, we'll call them samples for latent fingerprints. Are you just measuring the concentration of alcohol within sort of the sweat or the oils that are deposited with the fingerprint?
Yes, it very much is the sweat deposits that you find in fingerprints. If we start talking about fingerprints, your hands may become a bit sweaty because there is that response, and that's the fluid that we would be detecting our biomarker in.
Well, that makes sense. And I'd love to talk about your work in dry blood spots as well. Kim, I think this is an area that people are really excited about in general in anti-doping. So can you share your thoughts on what you see the role of these dry blood spots playing in the future of anti-doping?
Dry blood spots have a lot of potential in sports testing. As we know, they can be, again, collected more easily than having a phlebotomist collect venous blood, and they can be collected more quickly. So there is the potential for collecting in, say, a team event or a training event where you might want to do squad testing. So I think there are great benefits in terms of the number of tests that can be collected with a dry blood spot scenario. And again, because it's blood, the information that we get is relative to what's going on in the body at the time of the sample collection. So that, in a sense, provides some benefit when compared to urine. And there are some metabolites, so steroid esters is a good example, that are better measured in blood rather than urine. So dry blood spots and steroid esters, I think, is a good example of how that would be an innovation that would help the anti-doping field to have a better idea of drug use ongoing in the body at the time of competition or training or wherever the event may be.
Certainly. And you mentioned an ongoing area of research is being able to use these dried blood spots to detect growth hormone abuse to look for things like IGF-1. Can you comment on maybe where we're at with this type of question?
Many laboratories are looking to include dry blood spots in their repertoire of testing because of the points that I've just raised. We have particular expertise in growth hormone biomarkers, so it made sense for us to look at that particular biomarker. And the work is ongoing as we speak, actually. So we would then need to go through the process of validating that method and applying to add it to our scope of accreditation from the accrediting body. So the work is ongoing. It takes time to get to the end product, but it's certainly something that we would hope to be introducing into our testing portfolio sooner rather than later.
Oh, wonderful. Well, Kim, we are excited to see the results coming out on this work. I know we mentioned multiple times in our conversation the importance of collaboration and how you're working with a lot of different people to answer some of these important questions in anti-doping. So can you talk about, Kim, how you and your team collaborate with a variety of different partners, whether it's the Partnership for Clean Competition, the UK Anti-Doping, WADA, and other academic institutions to really tackle some of these big problems?
Working together is a key strength of the anti-doping world, I believe. There are annual meetings that take place, not least the one that's organised by the Partnership for Clean Competition, which are hugely valuable in bringing together laboratory directors, scientists, and other stakeholders to share good practice, to share research and innovation, and to try and work together collectively to take the field forward. So I think this is a really important aspect of what we do. We, of course, work with the national anti-doping organisations that are present in every country, particularly in the UK with our own, which is known as UCAD. We work very closely with UCAD to deliver their testing program, but also to share intelligence and to talk to them about how they would like to see the testing portfolio developed. So that kind of relationship is very important. We work closely with other funding organisations such as the World Anti-Doping Agency and share the good work that WADA and the PCC do in this area and try to support both to develop a knowledge base in anti-doping research and testing. So I think working together, collaborating is a key way forward, and it's a strength of the field as a whole. We, of course, work with other universities. We work with other faculties within our own university to collaborate and share research questions and undertake important research workshops in order to share the information and develop knowledge, not only amongst ourselves, but also amongst our PhD students and master's degree students who are, of course, the future scientists in this field. And I think importantly, we work very closely with other anti-doping laboratories. So there is an annual meeting of laboratory directors. There is a meeting of world anti-doping scientists. So meeting and sharing ideas is a way that we maintain our alliances and develop analytical and scientific wherewithal to address the anti-doping problems that we face, I guess.
Absolutely. And I think this sharing of information and working together is so important for getting that knowledge across and making sure we're not wasting resources with everyone sort of separately answering the same kinds of questions. So I'm really excited to hear that you partner with all of these different organizations and work together. And I'm curious, Kim, to learn a little bit more about how you first heard about the Partnership for Clean Competition and perhaps whether there are any particular memorable interactions so far.
Well, the Partnership for Clean Competition was already, I think, an integral friend of the Drug Control Centre. When I arrived here, I know that it's been instrumental in funding the work that we've done on growth hormone research, supporting one of our PhD students as a PCC fellow to carry on that work, and in particular work on the development of LCMS methodology for P3MP. So the PCC was here when I arrived, and we work together and support the work that it does as a continuing effort. Of course, the PCC actually came to King's a few years ago now to hold its annual research symposium, and we were able to meet everybody and again share the good work that you have been doing. So I think we have a very strong relationship with the PCC, and long may it continue.
Well, we appreciate you doing wonderful research and working with the Partnership for Clean Competition, like we said, to answer some of these big questions in anti-doping. And I think just moving the research forward is tremendously important for staying ahead of the cheaters. So can you talk about why you think it's so important to advance anti-doping science and clean sport, Kim?
I think anti-doping science is crucial for the integrity of clean sport. The testing program gives competing athletes confidence that professional bodies such as the accredited laboratories and WADA are serious about keeping sport clean. Working together and sharing intelligence is the way forward, and advocating that doping is at odds with healthy living, I think, is very important. So being in a university where I think well placed to advance anti-doping science, as I've mentioned, there is great interest from our PhD students and master's students in anti-doping science. So we're able to educate them on the importance of clean sport, and understanding that science underpins anti-doping is crucial, I think, to getting the message across about clean sport, and that we have the capability and the capacity from a laboratory analytical science viewpoint to challenge doping in sport and to enhance the integrity of elite sport.
Absolutely. I completely agree. And I think throughout our conversation, we've touched on or perhaps highlighted some of the challenges, some of the big questions that you're working to answer. But I know oftentimes resources are a limitation, unfortunately. So, Kim, if we gave you all of the resources you could dream up and there were no barriers, what is the one problem in anti-doping science that you would most want to solve?
Well, that would be very, very nice. I think that would be every laboratory director's dream. What I think I would spend the money on is testing. And I guess I would say that. I would spend the funds on establishing a robust testing program that ensured all athletes were tested regularly throughout the year. And that's not by any means to say that existing testing programs are poor. They're not at all. But I think, sadly, a lack of resources often means that elite athletes across many sports may not be tested optimally. Funding could support efforts to bring scientific knowledge to our testing programs. I think more could possibly be done to enable science to meet more regularly with policy, so that our collective understanding of elimination kinetics or urinary excretion of drugs on the prohibited list is enhanced, and that testing decisions or the testing program are endorsed by science. And again, that's not to say that that relationship isn't there, but I think it could be better. And testing at the right time and more frequently, whether this is a follow-up test for an APMU longitudinal monitoring or in the run-up to or after major competitions, I think could still be done better, and perhaps more so if it was done collectively with policy advisors and scientists getting together to talk about this.
Absolutely, Kim. I think you brought up some really important points here. And I really like the angle that you brought up of bringing together science and policy, because I think so often these kind of two areas operate in silos and we really need to make those bridges. So what are your thoughts on how these kind of two separate areas could be brought together perhaps more effectively?
Well, I think from the beginning, just having conversations, you mentioned the word bridges, and I believe that we could bridge the gap a little bit more by just having conversations or discussions, or when we meet at conferences or symposia, could we talk about testing? Could we talk about what our policy colleagues need to know about elimination of different drugs in urine, and whether we could provide that information to them that would help them frame their testing program? Does it need to be specific conversations about different sports or different classes of drugs? I just feel that conversations might take us forward some way in terms of feeling more confident that we're testing everybody that we need to test in the way that will enhance the integrity of the sport, and giving confidence to athletes that we're trying our very best to detect doping at an optimum period of time related to the drugs that we're looking at.
Well, Kim, I think that is an absolutely wonderful response there. And I think you're absolutely right. Bringing together people to have these conversations and to spark these collaborations, not just within science and within anti-doping, but across anti-doping and other areas, including policy, is critically important. And I know we covered a lot of different topics today. If we sparked the interest of our listeners out there and they would like to learn more, what is the best way for them to do so?
If they look for King's Forensics, and they can Google that, or directly put in www.kcl.ac.uk/forensics, they will find out all about what we do, including the latest interactions and work of the Drug Control Centre.
Perfect. Well, listeners, definitely check out these resources. And Kim, thank you so much for joining us on the show today. It's been a pleasure to chat with you.
And thank you so much again for inviting me to speak. It's been a thoroughly enjoyable experience. Thank you very much.
We appreciate it. And listeners, it's been wonderful to have you here with us as well. If you haven't already, it would mean so much to all of us if you could rate our podcast and leave a review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.